Well, thank you to Evan for that overview of the program. My name is Tom Wilson, and I'm the faculty lead of the program's core services support team. And for the next 10 to 15 minutes or so, I will be getting a little more practical on thinking about how to implement single cell and spatial analysis at the University of Michigan. So my objectives for this, my time here, is to first orient you to some of the strategic decisions that you might face when thinking about implementing single cell or spatial analysis tools in your research projects. To use that then as a way of thinking about where the resources exist and the infrastructure and services exist at the University of Michigan. And then along the way to give you some sense of our programmatic strategies for integrating, streamlining, and fostering collaborations between these multiple technologies and units. So to start out, we need to just understand that through what Evan was describing, single cell and spatial analysis is not just one technique. We're interested in the program in the integration from tissues that would give rise to dispersed cells that are analyzed in particular single cell ways, but also then being able to project those cells back into the tissues. And so it's maybe one conceptual idea in its whole, but it's implemented as different steps and technologies. And so you might, first of all, need to address for yourself the question as to whether you're interested in predominantly single cell or spatial data modalities. Single cell modalities take as their strength cell type definition, relatedness between individual cells, and examining heterogeneous samples for different types of cells, and in particular rare cells. Whereas spatial analysis really takes as its strength, of course, spatial location within tissues. Things like proximity between cells, the organizing principles of tissues, gradients, barriers, and the like. And these are not the same idea, and you may focus on one more than the other in your work. Now, the other thing is that we may not have to make a choice, because there are an increasing number of technologies that are starting to come out, which may allow us to actually look at more than one type of data modality at a time. That becomes true in analytes as well. So your second strategic decision would be whether you're interested in looking at RNA and DNA samples in your samples, or protein analytes in your samples. Nucleic acids are strongly driven by sequencing-based readouts, which are very powerful readouts that give high plexity and dynamic range, and of course are one of the only ways to really detect mutations as a direct readout. Whereas protein readouts tend to be antibody-based, and therefore can use a set of established markers that can be a great benefit to your study. And of course, they're the only way to get to definitive expression of proteins or to examine post-translational modifications. Again, this theme that we maybe don't have to choose so much anymore, because more and more technologies are coming out that are multimodal and might allow you to look at RNA and DNA, or protein cell surface markers and transcriptomes all in the same sample. The next strategic decision I'll emphasize is breadth versus depth. So do you want to look at more markers in fewer samples, or fewer markers in more samples? And here we can ask the question, how many markers does it take to really deeply characterize all of the cells in a tissue sample? And perhaps the answer may only be 20 or 40 markers, but there's a real challenge in establishing for any given tissue type which markers are the most useful. So you may find yourself over here on the left side, where you're really in the discovery mode of a study where omics applications will be better for you. As opposed to more mature studies, you might be looking at more translational applications with fewer markers to facilitate phenotypic correlations. The last strategic decision I'll start out with here is to emphasize resolution, which is a big one, because we have tissues here on the right where you're really going to be interested in things at the organ level of function, and therefore talking about larger biosamples. Whereas on the left side, we are also very interested in seeing spatial techniques applied at higher resolution, so that we're going to look at basic science studies of organelle function, for example. These are inherently going to be smaller samples simply due to the prohibitive cost of extending that level of resolution to really large samples. Now, historically, there's been a trade-off where the highest spatial resolutions were really only achievable with a smaller number of markers, and that as you move to the higher omics level data depths, you are inherently moving to lower resolution. We are increasingly seeing, and I think you'll see today, technologies that are allowing us to start to think about doing really the highest level of markers at the highest level of resolution. So again, maybe the trade-offs are starting to go away. All right, so those are the principles that will drive you into your studies and thinking about designing your studies. So how will you find them at the University of Michigan? So when we first started out in this program, we thought, should we create a new core facility? And the answer was quickly and obviously no, because there were already many sites on campus developing strengths in single cell and spatial analysis. So we see ourselves as more of a virtual core whose job is really to help integrate, foster communication, collaboration, to reduce redundancy between these sites, and so on. So what I'll do for the rest of my time is to just go around the block here and to introduce you to some of these individual facilities and what they're doing. So the Advanced Genomics Core is a core that I direct along with Olivia Hughes. And we've had for many years now a focus on single cell sequencing and mostly through the 10x Chromium platform with some of the applications indicated here. The asterisks throughout my talk will indicate that there is a short talk coming up immediately following me that will elaborate on some of these specific examples. In the last 2 years, we've been really seeing this single cell push expanding into spatial. So we are not only offering the 10x Visium platform, but we are one of the few certified service providers in the country, which gives us early access to things like, for example, the newest FFPE tissue processing capability. More recently, the program has helped purchase the NanoString Geomics platform, which we are now live on. And it has various features that, again, you'll hear about in a talk coming up. But I'll just quickly mention that it has a unique property of all the technologies and being really good at doing region of interest selection so that you can target your analyses. So moving on to the single cell shared resource in the Rogel Cancer Center, which Evan himself and Sunitha, who are part of our program leads, are the head of this core facility. And they've over years developed many sophisticated ways of doing single cell analyses. I'll mention that they also have a 10x Chromium that they run, and that the program purchased this Mission Bio Tapestry single cell PCR technology to allow DNA-based modalities at very high cell numbers. And as an example, one of the things that we're doing and helped by the program is to try to see how we might streamline this activity between this single cell shared resource and the Advanced Genomics Core. But the shared resource also has a focus on rare cell isolation through technologies indicated here. And Sunitha Nagrath will be talking about some of their work and with techniques such as the labyrinth pictured here. Great. So those are the omics first sites. Then I'll move to what I'm going to call the protein first sites. So the first is the CyTOF core led by Fei Wen. And here what we're going to see is a mass spectrometry based readout to look at either single cell or spatial analysis in the form of the Fluidigm Helios and Hyperion platforms. And this is 40 parameter mass cytometry, which is applied to detect either up to 100 proteins per individual cell in a flow type paradigm, or to use a tissue sample with 1 micron resolution and detecting up to the range of about 40 proteins in a sample. And again, you'll be hearing more about the Hyperion application in a short talk. That relates fairly closely to the tissue and molecular pathology core, supported again by the Rogel Cancer Center, but also very tellingly in the Department of Pathology, where Paul Harms, like myself, a pathologist, is applying a particular platform of spatial analysis. And here it's as a pathologist, I'll just point out that, of course, to some degree, this is histopathology. It's something we as pathologists have done for a long time. And so it's natural that such a site would expand into the Vectra Polaris multiplex immunofluorescence platforms, where we're having now smaller number of proteins, 8, that are being looked at, though, at increasingly high resolution, down to quarter micron resolution, leading to stunning views like this one that I think you'll see more of again in a short talk coming up. So let's now move to imaging first technologies and two core facilities that are both co-directed or directed by Nils Walter. The first is to just quickly mention the microscopy core. So of course, spatial analysis is in some very real sense imaging. So core facilities like Nils that have a focus on advanced imaging technologies are going to be very relevant. But these tend to not be marker associated, whereas the SMART Center, the single molecule analysis in real time group on the main campus in LSNA, also focuses on here STORM, superior super resolution microscopy, and then is coupling that with single particle and single molecule detections at the very much subcellular level. So again, here you can see very high resolution detection of single molecules. And Damon Hoff in the group is very interested in extending this to multiplex single molecule detections and techniques such as MERFISH. All right, so now we have to move to a different concept, which Evan clearly set up already, which is that everything we're talking about here generates big data and presents new and actually very exciting challenges and opportunities in data analysis. And that includes the fact that we have X, Y, and Z planes now with our sequencing data. We have to actually engage in this correlation of single cell to spatial analysis data modalities. We have multiple platforms. I think that's very obvious, and actually seeing those data play nicely together in single analytical platforms is a goal, among other things that I can mention, and I won't, because Arvind Rao is going to be giving a presentation on our efforts to marry activities through the Rogel Cancer Data Sciences group and the bioinformatics core led by Chris Gates. So again, you'll hear more about that in just a minute. And last but not least, we can't forget the roles of our research laboratories in supporting technology development as well as even delivery. So we have technology leaders such as Junhee Lee, and you'll hear from him at the end of the symposium about technology he's developing, or people who might otherwise be known as called early adopters, such as Sue Hamoud, who will be speaking. And then, of course, we are a faculty recruiting program, so we hope very much to put new recruits, developing still new approaches, and other members from already existing on our campus. And Soo-Min Lee is our newest member of the program, who will be talking about stunning images like this one. And that incorporates a component I haven't mentioned yet, which is lineage tracing over time. So not only what does the tissue look like now, but what is the pathway that it took to get there? Okay, so my last slide is to just emphasize that I realize this is complex. There are not only multiple technologies and opportunities across the campus, but even some of these even have multiple possible entry points. So there's different ways that you might prepare your sample or your tissue and get into these technologies. So it is very complex. And these advanced technologies clearly require a substantial investment of time and effort, not only on the part of the core facilities, but on you as well. And of course, these are expensive. There's a real cost to doing these, perhaps in the range of thousands, a few $1,000s per sample for some of these technologies. And so one of the things we have as a mission for the program is to create a pilot project program to help you get that pilot funding to get the preliminary data for your studies. All right, so that's what I had to say about orienting you in big picture to the campus. And now what we're going to do is turn to a series of talks from individuals who will follow up on a number of these different things that I've mentioned.